Guys, I'm Hosanna Barrett, a junior biology major at West Virginia University, and my project is Conidia-Corella luterostrata, a potential fungal biocontrol of the invasive Christmas tree pest elongate hemlock scale. I worked on this in the Casson lab in the WVU Division of Plant and Soil Sciences, and it was funded by the Christmas Tree Promotion Board. So elongate hemlock scale is an invasive insect that lives on the bottoms of fir, spruce, hemlock, and pine needles. And you can see it in the close-up here. It lives on the bottoms of the needles and sucks the sugars out of the needles, so it can have some negative effects, such as needle yellowing or needle drop. But in general, its main effect is that it's unsightly, and because of restrictions in moving invasive species, it can prevent the trees from being transported across state lines. And these are both an issue for Christmas tree farmers because they want to be able to grow and sell their trees. And elongate hemlock scale came over to the United States from Japan, and now it's spread across a lot of the East Coast. So you can see a lot of the spread in this map, but for example, in the inset showing West Virginia, they're showing it just in the Eastern Panhandle, and we found it in Mon County near Morgantown. So there's definitely some spread beyond what's recorded on this map. Hemlock scale is normally controlled with chemical pesticides, and those can be a good option, but they have non-target effects, such as killing the natural enemies of the scales. And so over time, sometimes the hemlock scale will rebound, and the population will actually be larger than what it was before because the natural enemies have been killed. So the life cycle of the scales, they're going to start out as an egg on the bottom of the needle covered by a waxy coating. And then the egg matures into a crawler, which is a mobile stage, so it can walk around. And then the crawler is going to mature either into a female adult, which is protected by the waxy coating the same way the eggs are and is not mobile, or the male adult, which can walk and is also has wings and can fly, although it's a weak flyer. But the most significant stage to us is the crawler stage, because that's when it spreads normally to other trees, and also it's the least protected because it doesn't have that waxy coating. Matt Casson and Brian Lovett from the Casson lab got this grant to look for fungi that infect elongate hemlock scale, and the fungi could perhaps be developed into a biocontrol agent to control the scales. So they went to Grayson County, Virginia, and Ashe County, North Carolina, to collect samples of Fraser fir branches that were infected with elongate hemlock scale. And they collected samples both from Christmas tree farms and from wild stands of Fraser fir. So once we'd collected the branches and brought them back to the lab, that's when I got involved with the project. And the next step was to inspect the branches and look for any visible fungal infection. So when we looked at the branches, it was immediately obvious that there was this fuzzy orange fungus that you see in the insert infecting crawlers on the branches. And this was widespread and visible even with the naked eye. So the next step was to grow this fungus so that we could do more research on it. So what we did is we just took the crawlers that were infected with the fungus, and the fungus killed the crawlers. So they were cadavers of the crawlers. So we took that cadaver and we scraped it off onto an agar plate so that it would have the nutrients that it needs to grow. And we grew several cultures that looked similar to that orange fungus that we'd seen on the cadavers. So the next thing we wanted to do was identify our fungus and make sure that all of our cultures were of the same fungus. So you can see in our cultures, there's a little bit of variation, and that's not uncommon with fungi just due to age and the growth conditions. So that didn't necessarily tell us whether it was the same or different fungi. But the next thing we did is we looked at the spores of all of our fungi in the microscope, and we compared them between the different plates, and we saw that they were similar. And we also saw this distinctive rice grain shape. And we compared that to journal articles, and we found that it was likely to be Conoideochrella luterostrata. So the next step was to sequence the DNA. So we looked at the DNA and we sequenced several genes of our fungus, and it came back as Conoideochrella luterostrata, just like we expected. And something that was notable is you can see it comes back similar to this strain from Kyoto, Japan. You'll remember elongate hemlock scale came over to the United States from Japan. And this strain of our fungus that was found in Kyoto was infecting scales there. So the fact that we found it in the United States infecting scales here as well is notable, and it means that it was likely imported to the United States from Japan, perhaps with elongate hemlock scale when they first entered the country. So our next steps, now that we have identified our fungus and confirmed that it infects elongate hemlock scale and that perhaps it's a natural pathogen that was brought with elongate hemlock scale from Japan, we want to do a bioassay on elongate hemlock scale or try to infect it at different life stages and see which stages can be infected and what the symptoms are of the infection. And we're also going to look for non-target effects. So those are if it infects other insects in the environment, such as the predators of elongate hemlock scale that we discussed, or if it has some effects on the plant when we apply it to the plant. We're going to look at that as well. And we also want to explore the diversity of the fungus that's present in the United States. So the sequences that we sent to be sequenced from the fungi we collected in Virginia, North Carolina, were all extremely similar to each other. But we know that in Asia, there's a wide variety. There's a lot of diversity of this fungus. So we want to know if there's only one strain in the United States or if there's diversity similar to what we see in Asia. And finally, the point of all of this is to formulate our fungus as a biocontrol agent so that it could perhaps be applied to the trees and used as part of a comprehensive pest management plan to control elongate hemlock scale. So that's the end of what we've done so far, and I just want to thank everyone in the Casson lab, and thank you for watching.